Uh, My name is Burwitz, and I work at Oregon Health and Science University in the Vaccine and Gene Therapy Institute. And our work focuses on simian immunodeficiency virus-infected macaques. And what we're trying to do is cure monkeys of SIV infection, trying to find a cure for humans for HIV infection. And the way we're doing that is we are trying to recapitulate the scenario of the Berlin patient, whose name was Timothy Brown, who is the only person on Earth that is known to have been completely cured of HIV. They've been unable to detect the virus, and so what we are doing is trying to recapitulate that situation to figure out how this happened and whether we can recreate it in the future in humans.
It's really, really exciting. In fact, I'm in love with your project. Can you tell me what your most exciting findings have been lately?
Yes, so the most exciting finding we've had most recently is that we can do these stem cell transplants. So the Berlin patient actually received a stem cell transplant from a person that was unable to be infected with HIV due to a mutation in the co-receptor CCR5. And the reconstituted immune system in his body is no longer able to be infected with HIV, and they believe that that is why he was cured of his HIV infection. And so we've recently obtained chimerism in our animal recipients, where we have transplanted stem cells from one monkey to another and have actually reconstituted the immune system of a second monkey with cells from the first monkey. So we're definitely closing in on our goal to try to recapitulate the Berlin patient. What I'm most excited to see here at IAS is really the coming together of so many different angles to cure HIV. So you have the latency reversing agents, the new, better, more specific and more neutralizing antibodies that are now being given passively to try to hunt down and reduce the virus in the circulation in HIV-infected people, but also the reversing of immune exhaustion with some different agents like TIGIT and TIM3, and I really think that a cure for HIV is really going to take kind of a combination of all these different areas of research to actually clear the virus from an HIV-infected person.
I agree. We saw that in cancer. No doubt we'll see this in HIV. Can you tell me how important amfAR has been in this work of yours?
Yeah, well, that's easy. So I think anyone who's tried to garner any sort of monetary research support knows that when you are a young PI and young in your career and kind of at the bottom of the totem pole, sometimes it can be difficult to find money, particularly when you're going for your first grant. I think once you have a grant, it might be easier to get additional funding, and I think what amfAR is really good at, and I know this because I know other young investigators that have been funded by amfAR, they really go out and try to find the people that are doing cool work but are still young, and so amfAR is playing a huge role in my career because they've given me my first grant, and I'm hoping this will be a launching point for the rest of my career. I'm actually very optimistic. I mean, I think being here towards an HIV cure symposium, I think it's really obvious, again, going back to this combinatorial treatment where we have so many different angles now on the virus, reversing latency, hunting down the virus after reversing latency, and then also reversing the immune exhaustion so that the cells can actually take care of those cells that have now had reversed latency. So I'm very optimistic that at least in a subset of patients that we will be able to cure HIV by 2020. Of course, something more broadly applicable to the entire world population, something like that's going to take a little bit more time, but I think we're really closing in on methods that will allow us to do it.